Okay, let's jump into this. Sperm. We think we know them, these tiny things responsible for reproduction.
Right.
But here's something I found really fascinating.
Yeah.
They actually need to be a bit cooler than normal body temperature to work properly.
Yeah, a few degrees cooler, typically.
Which immediately makes you wonder, well, how on earth do they manage to fertilize an egg inside the female body where it's obviously warmer?
That's the exact question, isn't it? It seems like a paradox.
It really does.
And that's precisely what this new study looked into. It's from researchers at Washington University School of Medicine in St. Louis.
Okay.
Published in Nature Communications. And, well, it's got some potentially big implications. Things like, future contraception or maybe even infertility treatments.
Right. Huge potential there. So our mission for this deep dive, then, is to unpack this research. How do sperm handle that heat and get the job done? What did the study find?
Okay, so the core finding is actually quite elegant. They discover that the warmer temperature itself inside the female reproductive tract acts like a specific signal.
A signal. What does it signal them to do?
It basically flips a switch in how they move.
A switch in movement. How so? What changes?
Well, normally they have this kind of smooth forward swimming motion, but when they hit that warmer temperature, they switch to this really energetic, almost frantic thrashing and twisting. It's called hyperactivation.
Hyperactivation.
And that specific kind of movement turns out to be absolutely necessary for them to sort of burrow through the egg's outer layers.
So they need that thrashing to actually penetrate the egg. The heat triggers it.
Exactly. Without that hyperactivation, fertilization just wouldn't happen.
So what's behind that switch? Like biologically, what's making it happen?
Right. So that brings us to a really key player, a protein called CatSper, C-A-T-S-P-E-R.
CatSper. Got it.
It's found in the sperm of all mammals. And its job is essentially to act as a channel, like a tiny gate.
A channel for what?
For calcium ions. These charged particles flood into the sperm's tail, the flagella.
Okay.
And that influx of calcium is what provides the power, the energy, for those really intense hyperactive movements.
So CatSper opens the gate, calcium rushes in, and boom, hyperactivation.
You've got it. That's the mechanism they identified.
And Dr. Polina Lishko was involved in this research, right? I saw her name. What was her main point about this discovery?
Yes, Dr. Lishko. She's a professor at WashU Medicine. She put it really clearly. She said something like, that hyperactive state is key for successful fertilization, and no one knew exactly how temperature triggers it.
Right, the trigger was the mystery.
Exactly. And she continued, our work has identified a temperature-controlled switch that triggers these increased movements precisely when they are needed. So it's about timing.
Yeah, it makes a lot of sense. You don't want them burning out too early. They need that burst of energy right at the egg.
Precisely. Conservation of energy is critical.
Now, I feel like I've heard other ideas about what activates CatSper before. Something about pH levels. Or hormones?
Yes, that's right. There were previous theories. One was about pH changes in the female tract. Another, mainly in primates, pointed to the hormone progesterone.
Okay, so why weren't those the full story?
Well, the progesterone idea, for instance, didn't really hold up universally because, as Dr. Lishko and others noted, the sperm of most mammals actually don't respond to progesterone.
Oh, interesting. So that couldn't be the main trigger for everyone?
Exactly. It meant there had to be something else, something more fundamental that worked across most mammals.
And that turns out to be temperature, which, thinking back to the start, kind of feels obvious now given their sensitivity.
An insight, right, but it connects beautifully.
It makes you wonder, though, why are sperm so sensitive to temperature in the first place? Is there an evolutionary angle?
Oh, definitely. There's a strong evolutionary reason. Think about how male reproductive organs are often positioned.
You mean like external tests in most mammals?
Exactly. That's a cooling mechanism. But there are other fascinating examples. Dolphins, for instance, use their dorsal fins to cool the blood going to their testes.
No way. Dolphins.
Yep. And elephants use their massive ears partly for the same reason, regulating temperature.
Wow. Okay.
And here's the kicker. Animals that don't have these kinds of special cooling adaptations for their reproductive bits, like birds, they also lack the CatSper protein.
Whoa. So the need for cooling and the presence of this temperature-activated CatSper seem totally linked evolutionarily.
It strongly suggests that, yes, the protein seems to have evolved alongside these cooling strategies as the final trigger for fertilization in the warmer female body.
That's really compelling. So, okay, how did the researchers actually prove temperature was the direct switch in the lab? What did they do?
They use some incredibly precise tools, micron scale stuff, actually techniques first developed for studying brain cells, funnily enough.
For brain cells
Yeah.
applied to sperm.
Yeah. These tools let them basically listen in on the electrical activity of the CatSper channel in single individual sperm cells.
Listening to electrical signals.
Tiny ones, yeah. And what they saw was really clear. Distinct electrical spikes showing CatSper was opening up, activating.
When?
Precisely when the temperature around the sperm went above 38 degrees Celsius. That's about 100.4 Fahrenheit.
So a very specific threshold.
A very specific threshold. They saw the temperature rise, saw the electrical spikes from CatSper, and then saw the sperm switch to that hyperactive movement. A direct cause and effect.
Wow, that's incredibly direct evidence. Okay, let's talk implications. The study mentioned contraception. How could this CatSper temperature thing be used there?
Right, this is where it gets quite interesting for potential applications. Because CatSper is only found in sperm.
So targeting it shouldn't affect other cells in the body.
Exactly. That's a huge potential advantage, fewer side effects. Now, people have tried before to make drugs that block CatSper, but it hasn't really panned out successfully.
Okay.
But this temperature discovery opens a totally different door. A new strategy.
Which is, don't block it, bud.
Activate it, but activate it too early.
Activate it early. How would that work as contraception?
The idea, as Dr. Lishko explained it, is maybe you could use temperature somehow to trigger CatSper prematurely. Get the sperm to switch into that energy-guzzling, hyperactive mode way before they reach the egg.
So they burn themselves out.
Essentially, yeah. They'd deplete their energy reserves. By the time they got near the egg, they'd be, in her words, powerless.
Like making them sprint the marathon warm-up lap.
That's a great analogy. Trick them into using all their fuel too soon. It's a non-hormonal approach, focusing just on sperm motility.
That's a really clever concept. Okay, what about the flip side? Infertility. Could this help people struggling to conceive?
Potentially, yes. If you understand the precise temperature trigger needed for activation.
You could maybe help sperm that aren't activating properly.
Exactly. Perhaps in assisted reproduction settings, you could carefully control the temperature to encourage that crucial hyperactivation step, maybe improving the chances of fertilization for some couples.
So understanding this fundamental switch could lead to tools for both preventing and promoting conception.
It seems possible, yeah. It addresses a key bottleneck in the process.
Okay, so let's wrap this up. This deep dive into the WashU study basically tells us that temperature isn't just a condition sperm have to tolerate. It's actually the trigger that tells them, okay, time to go into overdrive.
Right. It activates the CatSper protein,
which lets the calcium in, powers up the tail into hyperdrive mode, letting it penetrate the egg.
Precisely. It solves that long-standing puzzle about the temperature difference.
And it opens up these really intriguing possibilities down the line for new kinds of contraception, maybe non-hormonal ones, and potentially new ways to help with fertility issues too.
It really highlights how these, seemingly simple environmental factors like temperature can play such incredibly complex and crucial roles in biology. The elegance of the whole system is pretty amazing when you look at it closely.
It absolutely is. It makes you think. Considering this intricate dance between temperature and sperm at such a tiny level, what other basic environmental things, light, pressure, maybe tiny chemical gradients, might be holding surprising keys to other fundamental biological processes we just haven't figured out yet?
That's a great question. There's likely so much more subtlety in how life interacts with its environment than we currently appreciate.